In clinical oncology, there is a deeply entrenched baseline assumption regarding testosterone and the androgen receptor, or AR. Because of its well-documented mechanism in the pelvis, AR is heavily and almost exclusively associated with driving the progression of prostate cancer. But outside the prostate, applying that same biological logic fails. In different tissues, androgen signaling behaves in entirely unexpected ways. A robust body of literature now proves that AR signaling in non-prostatic malignancies is highly complex and profoundly context-dependent. Depending on the exact cancer type and surrounding microenvironment, higher androgen levels can either accelerate tumor growth or act as a protective shield. Treating the androgen receptor simply as a universal oncogenic on switch ignores the systemic reality of tissue context. This matrix maps out 3 distinct environments to analyze this hormonal behavior: breast, lung, and brain tissue. Here, context refers to intrinsic molecular subtypes of the cancer cells and the extrinsic tumor microenvironment surrounding them. We have to resolve a central tension in the data. Why does a hormone that rapidly drives malignancy in the pelvis actively suppress tumor progression in the brain? Predicting clinical outcomes requires mapping the exact cellular landscape. The androgen receptor is not a static trigger. It is a highly sensitive, context-dependent dial. In breast cancer, the impact of the androgen receptor is fundamentally dictated by the tumor's estrogen receptor, or ER, status. In ER-positive tumors, AR expression acts as a tumor suppressor, physically breaking growth. Patients with higher AR expression generally have a favorable prognosis, predicting better responses to hormonal therapies. Contrast this sharply. In ER-negative tumors, like HER2-positive and triple-negative cancers, AR functions as a tumor promoter. Because AR signaling is a poor prognostic factor in this context, current clinical trials are exploring AR antagonists specifically for these ER-negative patient populations. In breast tissue, the presence or absence of a neighboring receptor dictates whether AR acts as an oncogenic weapon or a protective shield. Lung cancer presents a different map entirely. Here, the evidence points to a unidirectional suppressive effect on tumor aggressiveness. Data displays a stark inverse relationship. Advanced lung tumor stages are consistently associated with lower AR expression. Molecular analyses explain why. The androgen receptor actively suppresses cancer cell invasion. Additionally, AR signaling alters the tumor, enhancing its sensitivity to standard chemotherapy drugs like cisplatin. The clinical data from the literature review confirms this protective correlation. In the lung, unlike the prostate, it is the loss of androgen signaling, not its presence, that signals terminal disease progression. Recent studies show that glioblastoma represents a striking exception to the general oncological rule that androgens impair our anti-tumor immunity. Recent preclinical data from the Cleveland Clinic identified a powerful site-specific tumor suppression mechanism in the brain. Looking at these timelines compares outcomes in murine models. Surgical castration slowed tumor growth in peripheral flank models, but in intracranial glioblastoma models, castration accelerated death. Pharmacological testing proved the effect. AR blockade via enzalutamide reduced survival, while administering exogenous testosterone rescued the deficit. Anatomical location reverses the physiological behavior of testosterone. A hormone that promotes tumors in peripheral sites becomes a critical neuroprotector. How does testosterone suppress brain tumors? In immunodeficient models, testosterone actually promotes tumor cell proliferation directly, but in immunocompetent hosts, testosterone's dominant effect is immune-mediated tumor suppression. This biochemical flowchart tracks the systemic cascade. Castration triggers hyperactivation of the HPA axis, elevating corticosterone levels. This severe stress hormone spike drives systemic T-cell exhaustion and dysfunction. Blocking the glucocorticoid receptor with mifepristone normalizes survival, identifying stress-driven immunosuppression as the core mechanism behind androgen loss pathology. Human registry data supports this. T-cell abundance declines with age in male glioblastoma patients, and men receiving testosterone replacement therapy for hypogonadism exhibit longer survival compared to non-supplemented controls. Androgen loss does not fuel the brain tumor directly. It disarms the host's systemic immune system, leaving the brain defenseless. These specific organ sites point to a broader implication. Varying androgen levels are a primary driver of sexual dimorphism in cancer susceptibility and treatment response. Androgens exhibit a biphasic response. triggering opposite cellular effects at low versus high doses through complex transcriptional and non-transcriptional pathways. This visualization models the tumor microenvironment. AR expression is not limited to the malignant cells. Androgen signaling manipulates the host immune cells within the entire microenvironment. This manipulation presents a highly tissue-specific avenue for overcoming resistance to targeted therapies and immunotherapies. Effective hormonal management now requires engineering the host's entire immune landscape, extending the clinical focus far beyond the simple starvation of a tumor. The glioblastoma data forces a severe clinical consideration. There is significant danger in the reflexive use of androgen deprivation therapies in male patients who are at risk for brain tumors. Combining low testosterone from androgen deprivation therapy with high glucocorticoid exposure, such as chronic dexamethasone, creates a compounding risk of catastrophic immunosuppression. Manipulating testosterone levels can alter immune cells within the tumor microenvironment. Oncology teams must carefully evaluate these interventions based on exact tissue context. The biology of androgen signaling has evolved. Relying on outdated assumptions about testosterone is clinically inaccurate. In certain microenvironments,